Thank you so much, all of the speakers, and I'm going to invite you all to go ahead and turn your cameras on. I'm Susan Pell, I'm the Deputy Executive Director of the U.S. Botanic Garden, and I'm going to moderate the panel discussion today. I would encourage all of our participants to go ahead and enter those burning questions that you've had while you've listened to these three wonderful talks in the Q&A, please. And we will get to them. And actually, we already have a few audience submitted questions. So I'm going to go through those in the order of the presentation. So Leonora, you've got two questions from the audience. One of those is, if a pitcher plant grows in a nutrient-rich environment, does it still produce functioning pitchers?
Yeah, so thanks for the question. There was a study that was carried out by Aaron Allison at Harvard Forest on Sarracenia purpurea, which is the local species there. And it was also a species that I was using in my study. And they found that when you added a lot of nitrogen, sort of as though it was nitrogen deposition, then those plants stopped producing pitchers. So they produced phyllodia, which are sort of flattened pitchers that don't actually have that cup and can't form a community. So it looks like, at least in certain situations, when there is extra nutrients in the soil, they stop producing those pitchers.
Great, thanks so much. And the next question for you is, are Sarracenia species classified as rare, threatened, and endangered in their native areas?
Yeah, so they are CITES listed, which is restricting international trade, but they are not sort of generally federally protected in the US, which is not great because they are hugely decreasing in terms, especially because of habitat destruction. So when I was sampling along the Gulf Coast, there were multiple sites that I visited where there were no longer Sarracenia pitchers, and it was kind of difficult to find those sites. A lot of times they're being drained because they're kind of boggy, so they're being drained for development. And then also things like nitrogen deposition could also be affecting these pitchers. So there are 3 species that are very restricted in where they are, and those are federally protected. And then there are some other states, individual states, that have regulations, but other states don't.
Thank you so much. We got one question for you, Dong. How many times has nitrogen fixation evolved across bacteria?
So first of all, nitrogen fixation is quite widespread among prokaryotes. So not only bacteria do that, but also archaea species can also have this ability. But if you look at the genes or the gene clusters required for nitrogen fixation, that's the nitrogenous enzyme and all the other associated proteins, they all look very similar. And the arrangement of this gene cluster is also very similar between different species. So they probably evolved only once and then just spread through rampant horizontal gene transfer between microbes.
Excellent, thank you. And Manu, we've got 2 questions for you. The first of those is, have lichen-forming fungi ever been found to steal genes from their photobiont? And is there selective pressure for the fungus host to evolve new growth forms to facilitate photosynthesis? 2-part.
These are excellent questions, yeah. And I think our knowledge about that, and I mean the lichenologist community, is very limited, and that is especially because of some methodological constraints, because of what other people have also mentioned here. You know, a lichen is a natural environmental sample that has all of these. So to be able to say with certainty which genes belong to which component, it's the ideal situation is if you have axenic cultures. So that's the only way you can say, oh, it is stealing or not. So up to my knowledge, I think the horizontal gene transfer is something that's very slowly growing in lichens, especially because of that. However, there are lichens that are being able to be cultured and the second separately. And then I don't think they have found these genes yet, but I could be mistaken. And the other question is about selective pressure. I think let me reread this.
I can read it for you.
To evolve new growth forms to facilitate photosynthesis. I actually never thought about that, because the lichens, they're always, evolving these. But what I mentioned is something that is very commonly seen in lichens, that the microbiome diversifies a lot while the photobiont didn't. And again, constraint, are they evolving new growth forms to facilitate photosynthesis, or, the photosynthesis is being performed by the photobiont? So it's a very gray area there. So it's hard to respond a yes or no question.
And then one more question for you. Is it common for lichen genera like Cora to be paraphyletic lineages?
Dictyonema that we've seen a paraphyletic grade, the sensu stricto, it is somewhat commonplace. I've been to discussions about these, and I guess it's the same as in other groups. There are people that are very sticklers to everything has to be monophyletic, to the people that also accept paraphily. Knowing this group now and working with this for over 10 years now, we've done different hypothesis testing, whether if it's paraphyletic or monophyletic, and we haven't found an answer that satisfies us. So given that the ecological and anatomical morphological forms still ties these groups together, I like to still accept them as a genus, even though they're not, looking so great now in their monophyly. So but I cannot say for my colleagues. I think it's in other areas is very variable. Thank you, Paul.
Great, thank you so much. And I have sort of a general question that any of you or all of you could answer. And that is, over the last 20 years or so, our understanding of how plants interact with each other, the environment, and other organisms has greatly expanded. Where do you think we are in the sort of discovery curve of understanding these dynamics? And where do you see your specific field heading? Leonora, maybe you can take it away.
Yeah, I can jump in. So I think it's really exciting. I mean, I think one of the biggest things that we've seen is that this recognition that these interactions, I think often people really talked a lot about competition and parasitism, but now we're recognizing that a lot of these symbioses can also be mutualistic, can be beneficial. And we're understanding so much more about the role of these environmental microbes that may not have this super tight one-on-one mutualism, but may also have a really, really important role in that system. So I think we're still on the forefront of it, really. I think that we're finding out so much more every few years about how these plants and microbes are interacting with each other. And I still think there's a lot to figure out about how endophytes and organisms that are coming from the environment are going to be affecting plants in a lot of ways, like in terms of avoiding herbivory, in terms of the secondary metabolites that they may be assisting in producing or sort of inducing, in terms of drought or temperature tolerance. So I think there's a lot of things that we're still going to be figuring out about how important these interactions are.
Awesome, thanks. Go ahead, Manu.
I can go next, and I couldn't agree more with Leonora. After seeing her talk, for example, I saw so many parallels in my talk, like working with completely unrelated lineages. And for example, she mentioned about convergent interactions, and that is something that I haven't been applied to lichens, for example. Even though we talk about these things, we see results. So I do think that symbiosis is a topic in general is something very exciting, and we still have a lot to be discovering. And so I agree, we're at the beginning of the curve, and I still believe that we're doing the best to what's available out there, but new technologies, new methods, new analysis are gonna keep coming, and we'll be able to further explore this. But I think all along is very exciting times.
Excellent, thanks. Dong, do you have any thoughts about this question?
Yeah, to me, for what I study, I think the really powerful tool is plant genetics, not only in legumes, but also in non-legumes that form very similar nitrogen-fixing symbiosis with other microbes. So we are discovering so many genes that are participating in this process. And also, it seems that every other gene we discover is also required for the arbuscule mycorrhizal symbiosis, and just shows you how closely related these two mutualistic interactions are. And I think then we're getting close to understanding why, for example, legumes, among other many other plants, have this rather unique ability to engage with nitrogen-fixing rhizobia, and what's the trick that, if you like, that legume developed. So I think we're close to understanding this question now.
You've actually got an audience question that's just come in, and that is, it says, you mentioned that nitrogen-fixing symbioses evolved from arbuscular mycorrhizal symbiosis within the angiosperms. Is there a likely reason why the bacterial symbiosis has not evolved in other groups?
Right, and that's the million-dollar question, right? And not only in ferns and gymnosperms, but also even within angiosperms, right? So monocots, for example, do not have this, at least do not have the one that involves building a nodule where you keep the bacteria inside your body, so to speak. So we don't really know. It's possible because at least efficient nitrogen fixation requires very intimate interaction between the host, and that's best achieved by engulfing the bacteria into the host cytoplasm. And it's possible that other species of plants simply have not figured out how to do that without compromising their very powerful immune systems. Right? So that's only a speculation. But in support of that, we know that most microbial species that interact with plants, they do so from the extracellular space. Yeah, and that's in contrast to animal system where a lot of pathogens go inside the host cell, both bacteria and sometimes fungi can go into the host cell to establish a niche intracellularly, which you would think is a very nutrient-rich environment. So why do plant-associated bacteria do not do that? So that's the question, and that's possibly the answer.
Excellent, thank you. And Leonora, you have a new question as well from the audience. What types of microbial communities are associated with other carnivorous lifestyles, such as sticky traps?
Yeah, thanks. There was a paper a couple of years ago that was looking at Venus flytraps and looking at the microbes that were present sort of at the different stages of the trap being open and closing. But as far as I know, we don't really have a good sense of the microbes that are associated with sundews and other sticky traps. And I think it'll be a really interesting area to keep studying. I know some people that are potentially working on that now. And yeah, you may see that there are organisms that are sort of attracted by some of the sugars that can be present in those sticky substances, in the same way that the insects are probably attracted to that too. So I think it'll be interesting to understand more about if there might be microbes that are playing a role in that digestive process as well.
Very cool, thanks. Manu, I have a question for you. Lichens are remarkable in so many ways and are often found growing in locations and environments where other organisms either really struggle to survive or just cannot survive. I wonder if you can talk about how the symbiosis between organisms and within lichens makes it possible for them to survive growing in these extreme environments like in Antarctica and on really hot rock surfaces, that sort of thing.
Thank you, Susan. Yeah, I often, well, I'm very biased to say the lichens are amazing, right? But I agree. I think lichens have evolved to be in places where no others. And there is research showing that can be for a variety of reasons. So for example, of course, adaptation, but also lichens also have different acids and pigments. And some of them might adapt, for example, if you are if the lichen is exposed to very intense luminosity. So we think, oh, it's nice to go to the beach, right? But imagine living there constantly. So lichens have adapted and they produce certain pigments that act as a sunscreen, for example. Other reasons are the microbiome. So there is one study that compares, and that's another part of the talk that during Leonora's that it made me think of this paper, is that they tested the microbial communities in this Citaria culeata lichen, and then they found that the ones living in, for example, Antarctica and other cold environments were more closely related. These microbial communities were more closely related or similar to those of the same lichen but living in the Mediterranean. So I do think that we still have a lot to explore in the microbiome, because they could be helping and facilitating and evolving with certain lichens to adapt to such environments that are considered to be so extreme for others.
Great, thank you. And I can speak for a vascular plant scientist in saying that you should be biased. Lichens are amazing. They're fantastic. So thank you for studying them. This is an audience question for everybody. Often scientists may mimic processes in nature for human inventions. Do any of your research or of plant communities and symbiosis remind you of how human communities interact?
I can jump in. I do think about the parallels with human communities. And I think in the little pitcher plant microcosm, you have all these different organisms that are carrying out different roles. And so it is a little bit like a society where different people have sort of different roles within that society, or maybe different personality types and things like that. And they tend to play different roles within those interactions. But that you may see that happening over and over in many different places, right? That each different city or town or country may have different people that fill those similar sort of niches or roles within communities. So I like thinking about it in that sense.
I can jump in also. I was mostly not thinking about how human communities interact with each other, but how human society interact with the world, with the natural world. So the way that a legume plant would go out and capture a symbiotic microbe, sometimes even one single cell, and then turns that essentially into a nitrogen fixing worker or colony of workers for it. It reminds me of how human societies would go to the wild to, for example, to capture an elephant and then train it, tame it, and make it into a livestock. So that's why I think I make the analogy between, use the word domestication, where in this case the plant is the active individual that domesticates bacteria for its purpose.
That's great. Manu?
Yeah, I can just complement to that. And I once heard this from my postdoc advisor, one of them, Martin Grube, and he sort of explains this lichen symbiosis as something very similar to what my colleagues already said. And I don't want to exclude any lifestyle, but for example, you are the microbiont, and then you have a partner, which would be the photobiont, and the two of you were primarily, shape this symbiosis. However, you have friends and family, which in case, or the community, which are the bacteria and other fungi, that will sort of supplement to that. So the lifestyles, the things that you like to do, these are going to complement, but the home is where you and your partner are. So again, not trying to exclude any, like the single lifestyle, but an often analogy for the lichen. So when you see in textbooks too, there are some analogies for that as well.
Excellent, thank you. I loved all those answers. That was great. I'm wondering if we can talk a little bit about sort of obligate associates and those that can be free living, and just sort of put that out there just kind of broadly. And I think one thing in particular that I'm interested in is sort of, are there any common characteristics of obligate organisms where maybe they've lost something in their genomes, or there's some kind of common characteristic, and really kind of looking maybe even across organisms in that area. I don't know, Leonora, do you want to jump in on that one?
Yeah, so in the pitcher plant system, we don't know if any of the microbes are obligate or not, because they're likely, the pitchers are generally thought to be sterile or very close to sterile when they open, and then they are colonized from the environment. But you do see that some of the pitcher plant mosquitoes or midges, some of these dipterans are really closely associated with their host, and you don't find them in any other environments. So even though they are sort of horizontally transmitted, right, from pitcher to pitcher instead of passing down through a lineage, they do seem to keep that very tight relationship. And that could be true for the microbes too, right? It's just harder to know because it's harder for us to really say, we don't find these microbes anywhere else. They're only in the pitcher plant, right? And I think that for some of these microbes, it may be that you could find them in the soil, but they might be dormant. They may not be really active, and they might be waiting until they get into their habitat where they're more happy to be. So in that sense, you could have a relationship that's kind of between, it's in between the obligate and the sort of more ubiquitous associations. We definitely know from symbioses, especially between insects and microbes, that they often do lose genes when they become obligate, and it sort of they end up having the microbes end up having smaller genomes, and they become more tied to the reproduction and the lifestyle of their host. And we don't see that in our pitcher plant organisms, but I think it's a really interesting question, and I want to understand it more, and to what extent, even if you find the organisms in other habitats, that this may really be the location where they're thriving and actually sort of reproducing and duplicating and more metabolically active.
Yeah, so if I can add, then in obligate system, you often see that one of the partners, they lose some essential metabolism, right? And then also their DNA repair pathway is degraded. They do not repair their DNA very efficiently, and then eventually their genome just degrade because they have much smaller genomes. But if the association is not obligate, then the organism can have a different lifestyle. Then very often you see actually the genome is actually bigger than the otherwise entirely non-symbiotic relatives. For example, rhizobia compared with other bacteria, has a pretty big genome. That's because it needs to live inside the host, inside nodules, but also you need to be able to survive in the soil for years when the right host does not come across. So that's just from the genome point of view, they're very different.
Mm-hmm. Yeah, I can just briefly mention this for lichens too. I think finding for example, I briefly mentioned that there are ascolichens and basidiolichens, and finding out now there are a lot more ascolichen genomes available. I'm generating some of the basidiolichens, and I think comparing to see if there are genes that are present in lichenized lineages that is lacking in non-lichenized, because for example, the group that I work with, its closest relatives are not lichenized. So seeing which genes are present or not. And something else is about the functional diversity of these bacterial communities in the microbiome side of things. So we found that the microbiome of basidio and ascolichens is very similar up to the order level or family level, but then under it, so the genes for the genera in Rhizobiales that have been found in both were different. So they are not potentially the same, taxa present, but the function that are performing in basidiolichens versus ascolichens are similar. So I think there's a lot to be done there still, but we already started to see some of these patterns.
Oh, so fascinating, all of it. This is sort of a kind of a fun, open up question here. What's the most surprising thing that you have discovered about plant symbioses or mutualisms in your systems?
I think that when I first started studying these types of interactions, I thought that they were more black and white, right? That like something was either a parasite or a mutualist. And I think as I studied it more, I just realized that they're all on a spectrum. And so a lot of times it's context dependent, and something in one context can be a great mutualism, and in different contexts it can be a parasitism. And so sometimes those terms are not as helpful as terms that are really describing more the actual interaction and the trade-offs and how those might change under different contexts. I think that's something that I found surprising about symbiosis.
Yeah, I just want to second what Leonora said, that mutualistic interactions in my field, I found this the surprising discovery is that mutualistic interaction is also antagonistic, but it really shouldn't be a surprise in that, because we would not understand that unless we consider evolution, right? Because nothing makes sense unless you consider the evolution force. And even in mutualistic interactions, no species exists for the benefit of other species, right? So if they are able to take advantage of their partner, even though it's nominally a mutualistic interaction, one species will do that. So we really shouldn't be surprised when we found out that even in a working mutualistic relationship, there's also this competition between the two organisms.
Yeah, and I think with lichens, we've probably heard many times that they are mutualistic, but there's an old idea by Ahmadjan that already mentioned that lichens could be a controlled form of parasitism. And now the more broadly accepted idea is that each case is a case. So with lichens, it's very hard, there's no one size fits all. And with lichens, I think we're almost constantly challenged by the way that the new discoveries. And I feel like it's learning grammar. You feel like you finally had a grasp on something, and then there's like another exception to the rule. So I think this keeps lichens really, really exciting, because it's, even for a definition of what a lichen is, there's a lot of discussion and disagreement in the community. And I think that's very challenging for all of us, but at the same time, very exciting to keep these discussions open and these conversations. So
this is maybe a little bit more difficult to kind of tease out, but I'm wondering, for those of you who study systems where you have, they're non-obligate participants in this sort of collaboration or mutual parasitism, however you want to talk about it. Is there, do we know what kind of the impact on the sort of function, the ecological role, the sort of environmental actions of those organisms, or interactions rather, with those organisms? What impact the sort of being in the mutualistic or symbiotic relationship has on their function versus if they're growing on their own? Like, how do they change kind of function-wise? I don't know, Leonora, do you have any thoughts on that?
Yeah, well, people often ask me if I think pitcher plants could grow without their microbiome. And I think they probably could, in the same way that you can rear notobiotic mice, right? Mice can grow without a gut microbiome, and they're okay, but they're often missing important sort of parts of their functioning, right? They're not completely entire organisms because they're missing this other whole community that's actually part of that organism. So I sort of think of it in a similar way. And pitcher plants often do produce some of their own digestive enzymes. So they produce proteases, some even produce some of their own chitinases. So I think that they could still break down some of that prey, but they wouldn't get the full spectrum of the nutrients that they might be able to get with all this additional metabolic activity that comes from their microbial communities. So yeah, so I think of it sort of like a gut microbiome, I guess, where you could probably still digest your food okay, but not everything. And you'd be missing out on a lot of nutrients that you could be getting from other foods.
I love that analogy. That's awesome. Anybody else have some thoughts on this one?
Right. So for the systems that I study, it's quite obvious. It has huge impacts, both to the environment, both the natural one and also human environment, since we use so many legumes as our crops. And that symbiosis made both rhizobia and legumes very, very successful, immensely successful. It seems that for rhizobia to enter this symbiosis, you didn't lose too much of anything. But for legumes, one of the open question is that once you engage so intimately with the bacteria, so each legume cell in the nodule can have hundreds, if not thousands of bacteria. Those host cells are essentially bags of bacteria. How can you protect yourself from bacteria, the rhizobia taking over all other bacteria that may come in and sneak in as parasite or pathogens? So what kind of immunity changes in immunity in the legume genome or legume function that has to be made to accommodate this kind of symbiosis? We really actually do not know. But we know the genes that are important for symbiosis. We know the genes that are important for host immunity. So we can put the two together and try to figure out.
Yeah, I have a lot of thoughts about this. I'll try to be concise. For example, I think this is part of the dilemma right now, is to define who is really important for these and who is not. And then again, this comes with a lot of limitations, because where do you draw the line of who's a partner and who's not? So for example, some of these bacteria, it's very hard to produce a lichen in the lab, right? Because in theory, if you have only two things, you separate, you culture, you put them together, they will grow. But for a lichen, in simple words, it looks like a blob. It doesn't look like a lichen thallus. So in that sense, it would make sense that if this microbial complex, bacteria and fungi and archaea could be present, and if they're not, then they're not going to create this. And on the other hand, going back to your question, it's I think one way that we can see this is testing what are the same lineages, especially in green algae or in cyanobacteria, that are able to live outside of the thallus versus the ones lichenized, and then compare what genes have evolved for that and to see if it's beneficial or not. But again, it's very, at some level, difficult to find exactly these lineages to test, because we still don't have very well established species boundaries, not only for the microbiome, but for the photobionts as well. So making sure, again, that we're comparing apples to apples is the first challenge that we have to pass. So creating this space knowledge is very important.
Yeah, I totally agree. Taxonomy being the foundation of so many things. So wonderful. I want to thank you all so much for these 3 fantastic talks today, this really engaging panel discussion. I'm going to wrap up the panel discussion with our final question, which is the same one I asked the panelists yesterday, and that is, in the spirit of a 30-second elevator pitch, you can take a little more time than that if you want, what is the most fascinating aspect of your research, or the thing that you really want everyone to know about the organisms or the system that you study? We're going to start with Dong.
So I think, the one that I come back thinking about after studying this is that in the nitrogen fixing symbiosis, at least in legumes, nitrogen is really the key, and it's probably key to many of the questions that we don't really know the answer to. It sounds simplistic, of course, but if you compare the nitrogen fixing symbiosis in a nodule on a legume tree, it's very unique. It's very strange. Right? In that the host produces a dedicated organ, engulfs bacteria, and then also sends a lot of antimicrobial compounds, peptides, to the bacteria. And it doesn't quite make sense unless you think what the need of this symbiosis is meeting is basically to make nitrogen. So what are the chemical needs that will allow nitrogen to be made? And also what are the needs for the plant to retrieve those nitrogen from the bacteria? So all those characters make sense if you put nitrogen into the picture.
All right, thank you so much. Manu?
Well, I think I've conveyed my message before, but since I have the floor, I'm just going to go with, I think lichens, it does take a village. So a village of people that we need to work in collaborations. And there's just so many lichens out there, so many unknown interactions. So the more people that we have working together, we're able to then go across different ideas and evolve or advance the knowledge that we have, because these are very complex symbiotic systems. We have a lot of them, not a lot of lichenologists. And on the other side of the story that it takes a village, it really takes a village to make a lichen as well. And I think it's one of the most unique symbiosis, because you only see the symbiosis when it's together, right? So one of the classic examples, of Finding Nemo is the clownfish and the sea anemone, or trees and mushrooms. You see the symbiosis, but they're two different organisms. And in the lichen, all you see is one structure. So I think that makes lichens very unique, and there's just so much unknown. So it's a plea to involve people in lichen education everywhere. So if you have a class or if you have a talk, make sure that you include lichens as an example.
Wonderful, thank you. And Leonora.
I think my takeaway message is really that we can study things at different scales, and it's important to be able to look at those different scales. So when we zoom back and we look really broadly at patterns, it helps us to understand sort of the basics, the fundamentals of how things come together, and sort of why we may see certain processes happening in the world, certain patterns over and over, right? So convergent interactions is really that idea of stepping back and saying, if you look really broadly across life, do you see these same kind of interactions happening over and over? And can we use those patterns to understand more about how life works? But then you also see that it's really important to study the details as well, because when you zoom in on something like these unusual plants or on lichens, you just see that there is also so much complexity. And if you only look at the big broad patterns, you miss all these important details that are also really essential for understanding how life works. So I think that my main takeaway message is just that both of those perspectives are needed to be able to see things broadly, to look at these patterns that we see repeating over and over, and then also to zoom in and look at the details and to really understand what makes things unique.
Wonderful. Thank you all so much. And I really want to thank three speakers today, the three speakers from yesterday, all the wonderful audience members and the engagement that we've had in these panel discussions. And I want to invite everybody to send symposium topic ideas to me. I'm just putting my email address and name in the chat there, so you can pull that. We do these every year, and we're soon going to pick next year's topic. So please send us your ideas, and we look forward to seeing you next year at the 19th Botanical Symposium in partnership with Smithsonian's National Museum of Natural History's Botany Department and the US Botanic Garden. Hope you all have a great rest of your day.
Thank you, Susan, for moderating. And thank you, Manu and Dong, for your talks.
Thank you very much.
Thank you so much.
Bye-bye. Thank you.
Bye.